Hello and welcome to our video. Today we're going to talk about the recombinant DNA. Recombinant DNA is a technique for cutting and pasting DNA sequences of interest using enzymes. The recombinant DNA sequences can be inserted into vectors, which transport the DNA to a suitable host cell where it can be replicated or expressed. They are DNA molecules created through laboratory methods of genetic recombination, such as molecular cloning, to combine genetic material from numerous sources, resulting in sequences not found in the genome otherwise. In our DNA technology, the first step is to isolate the DNA in its purest form, because DNA interacts with other macromolecules such as RNA, polysaccharides, proteins, and lipids within the cell membrane must be separated and purified using enzymes such as lysozymes, cellulase, chitinase, ribonuclease, and proteases. Other enzymes or treatments can remove other macromolecules. Restriction enzymes. Restriction enzymes cut DNA at certain sites like molecular scissors. They include incubating pure DNA, the restriction enzyme of choice, at conditions that are appropriate for that enzyme. PCR. PCR is a method of making multiple copies of DNA sequence using the enzyme DNA polymerase in vitro. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. PCR reactions are run on thermal cyclers using the following components: template, primers, DNA polymerase, and nucleotides. Ligation. Ligation of DNA molecules is the process of putting cut fragments of DNA and the cut vector together in the enzyme DNA ligase. The resulting DNA molecule is a hybrid of the interest molecule and the vector DNA molecule. As a result, this new hybrid DNA molecule is known as a recombinant DNA molecule. Next, we're going to talk about the insertion of recombinant DNA into host. In this step, a recombinant DNA is introduced into a recipient host cell, mostly a bacterial cell. This process is transformation. Bacterial cells don't accept foreign DNA easily. Therefore, they are treated to make them competent to accept new DNA. Then we're going to talk about the isolation of recombinant cells. Isolation of recombinant cells is the transformation process which generates a mixed population of transformed and non-transformed host cells. The selection process involves filtering the transformed host cells only. For isolation of recombinant cell from non-recombinant cell, marker gene of plasmid vector is employed. Now we're going to talk about the uses of the recombinant DNA. Recombinant DNA is used in vaccines and protein medicines such as human insulin, interferon, and human growth hormone. They all benefit from recombinant DNA technology. It's also employed in the development of gene therapy and the production of clotting factors for the treatment of hemophilia. But how is hemophilia treated? Hemophilia is treated by the factor VIII replacement therapy. Factor VIII replacement therapy has been improved through increased knowledge of molecular mechanisms regulating blood coagulation, derived largely from the isolation of the factor VIII gene and its expression in mammalian cells. Homogeneous pure preparations of the factor VIII, the largest, most complex protein, pharmaceutical produced today through recombinant DNA technology. Herbert Boyer of the University of California in San Francisco, Stanley Cohen of Stanford University were the first to produce recombinant DNA in a living organism in 1973 using E. coli restriction enzymes to insert foreign DNA into plasmids. Thanks for watching the video and goodbye.